Hello everyone! Today we're going to be talking about Chapter 29, Plant Diversity Part 1, How Plants Have Colonized the Land. Great title! Let's begin with learning how land plants evolved from what we call as green algae. So, first reporter, take it away.
Good day everyone. Today we are exploring the origins of terrestrial life by looking at concept 29.1, how land plants evolved from green algae. We know plants are related to algae because they share several fundamental traits. They are multicellular, eukaryotic, photosynthetic autotrophs with cell walls made of cellulose. However, the group of green algae called charophytes are the closest relative to land plants. We can see this link in unique structures such as rosette-shaped protein rings that synthesize cellulose and similar flagellated sperm. We can also see this in peroxisome enzymes and also the formation of a phragmoplast. The question now is, why leave water? Well, because the new frontier was spacious, and also plants found unfiltered sunlight and an abundance of CO2 and soil rich in mineral nutrients. So there was more opportunity for this organism to grow and develop. Okay, but this move came with a major risk: desiccation, or in other terms, drying out. Natural selection favored individuals that could survive periods of drought. This is where sporopollenin comes in. In charophytes, this durable polymer protects zygotes from drying out in shallow water. In land plants, this same chemical adaptation encases spores, allowing them to be dispersed through dry air without being harmed. While plants share traits with algae, they have four derived traits that are absent in charophytes. These were essential for surviving on land. The first is alternation of generations. The second, sporangia that produced walled spores. Third is gametangia that produce gametes. And fourth is apical merisms, and I'll be exploring these individually in the following slides. Okay, the first one is called alternation generations. Plants alternate between two multicellular stages, the haploid gametophyte, which produces gametes by mitosis, and the diploid sporophyte, which produces spores by meiosis. The fusion of gametes, male and female, gives rise to a diploid sporophyte. This, in turn, produces haploid spores by meiosis, and the diploid embryo is retained within the tissue of the female gametophyte. The parent transfer cells are cells that transport nutrients from parent to the land embryo. Land plants are called embryophytes because of the dependence of the embryo to the parent. The next key trait is sporangia that produce walled spores. The sporophyte stage has multicellular organs called sporangia. This is basically the organ that produces spores. Sporocytes, diploid cells, undergo meiosis to generate haploid spores. The spore walls contain sporopollenin, which we discussed earlier, and this makes them resistant to harsh environments. This is a cross-section of a sporophyte, or specifically a sporangium, and you can see spores inside. The next derived trait is gametangia that produce gametes. Gametangia is basically organs where gametes are produced, and there are two types. Archegonia is the female gametangia. It produces eggs, and it is also the site of fertilization. The male gametangia is called antherdia, and it produces and releases sperm. The last derived trait of land plants is apical merisms. Plants can sustain continual growth in these apical meristems, and they can be found on growing sites such as the tips of shoots, the tips of roots, and anywhere where growing happens. And these cells basically specialize. The last part of this concept is the origin and diversification of plants. Life on land, as I said earlier, began roughly 470 million years ago. We know this because we've extracted fossilized spores in tissues from this date, and those ancestral species gave rise to the diversity of modern plants. We can see on this slide fossilized spores, and fossilized sporophyte tissues. And that's it for this concept.
So now, let's move on to the next concept, which focuses more on mosses and other non-vascular plants. Scientifically known as bryophytes, these are plants that do not have a vascular system. There are three main groups. Notably, they are mosses, liverworts, and hornworts. Now, it is worth noting that phylogenetic analysis indicate that these bryophytes diverged from other plant lines early in the evolutionary history, as illustrated in the phylogenetic tree shown in this slide. So next we have the life cycle of a moss. But before that, it is worth noting that plants alternate between two very distinct generations, the gametophyte stage and the sporophyte stage. For bryophytes, the gametophyte stage is the dominant stage and it makes up most of the moss's structure. So with that being said, we can start with the first segment, which is spore germination and protonema formation. So to start, the moss cycle begins with a haploid spore. When it lands and grows in a moist environment, it germinates and grows into a thin, thread-like structure called protonema. This protonema then produces small buds through mitosis, which will develop into the mature moss plant. So now we move on to our second segment, which focuses more on gametophyte development and reproductive structure. These buds grow into leafy gametophytes, which are anchored to the ground by something we call as rhizoids. Now comparing them into roots, rhizoids are far more simpler as they don't have vascular tissue, which means they don't absorb much water or nutrients compared to roots. Once the gametophyte is fully developed, it forms reproductive structures called gametangia. The male structures are called antheridia, and the female structures are called archegonia. So now we move on to the third segment, which focuses on fertilization and embryo development. When water is present, the sperm from the antheridia swim through a thin film of moisture toward the archegonia to reach the egg. Fertilization happens here, forming a diploid zygote. This zygote then stays inside the archegonium and develops into an embryo through mitosis. So now we move on to the fourth segment, which focuses on sporophyte growth and spore release. The embryo grows into a sporophyte, which remains attached to and dependent on the gametophyte for nutrients. It develops a stalk called a seta and a capsule. Inside that capsule, meiosis occurs to produce haploid spores. These spores are then released into the environment, starting the cycle all over again. Even though mosses, liverworts, and hornworts have been classified under as bryophytes, they still have some differences, which we are going to be talking about in biodiversity. So starting with liverworts, there are actually two types. There are thalloid and leafy. So for an even simpler explanation, thalloid liverworts have flat, soft, ribbon-like plants. They don't have real leaves or stems. They have simple internal spaces and small openings, like a primitive stomata, to exchange air. Meanwhile, leafy liverworts are much more common and look a little bit like mosses. They have quote-unquote leaves arranged in 2 or 3 clear rows, and these leaves often have rounded edges that look flat and delicate. Now we can actually move on to talking about the liverwort's gametophyte and sporophyte. So starting with its gametophyte, it produced umbrella-like stalks like archegoniopores and antheridiopores to house their reproductive organs. Their sporophytes, however, are quite modest as they consist of a short, translucent stalk called a seta and a simple capsule that splits open into 4 valves to release spores. So now let's talk about mosses, which are the most diverse and widespread among the bryophytes. So in terms of its gametophyte, it is typically arranged spirally around a central stem. They also have a distinct midrib that provides structural support, allowing them to grow slightly more upright. In terms of its sporophyte, they are much more persistent and visible. They consist of a long, sturdy seta and a more complex capsule topped with a caltra. Most mosses also feature a peristome, a ring of teeth at the capsule mouth that opens and closes based on humidity to precisely time spore release. So last but not the least, we have the hornworts, which I consider the most unique. among all the three bryophytes. So in terms of its gametophyte, it is a thin, flat blue-green thallus that grows horizontally along the ground. They also share a unique characteristic of housing nostoc, a nitrogen-fixing cyanobacteria which provides nutrients to the plant. In terms of spirophytes, named for their horn-like appearance, these spirophytes are elongated green cylinders. Unlike mosses or liverworts, hornwort spirophytes lack a seta. Instead, they have a meristem at the base that allows the horn grow continuously from the bottom. So before we move on to the next concept, it is worth noting that mosses have a significant ecological and economic importance. Ecologically, mosses help maintain nutrient balance in ecosystems by reducing nitrogen loss. An certain experiment demonstrated that soils with moss retain over 95% of nitrogen inputs, versus soils without it that lost so much more through leaching. Economically, peat derived from moss has been widely used in many aspects of agriculture, forestry, heating, and etc. And it shows that to this day, it still remains important to some regions.
Hello, so I will be discussing the last concept of this chapter, concept 29.3, ferns and other seedless vascular plants. But first, we will be talking what vascular plants are. Vascular plants are plants that were among the first to grow tall on land because they develop vascular tissues that transports water and nutrients efficiently. This adaptation allowed them to become larger and more complex, marking an important step in evolution. Seedless vascular plants appeared around 425 million years ago, but unlike earlier plants, they developed specialized tissues called xylem and phloem, which helps in transporting water, minerals, and nutrients. Because of these features, they were able to survive and thrive on land. Vascular plants are characterized by having true roots, stems, and leaves, dominant sporophyte generations, and the ability to grow taller to compete for sunlight. These plants undergo alterations of generations. The sporophyte is the dominant stage that produces spores, while the gametophyte produces gametes. One example of a vascular plant is a fern. So, we will be now talking about the life cycle of a fern. First is, spores are being produced by sporophyte through meiosis in the sporangium. Spores are tiny single-celled reproductive unit produced by seedless plants to reproduce, while sporangium are microscopic specialized reproductive structures found in plants that produces spores. The second is, spores will then turn into a gametophyte which produces haploid male and female reproductive cells called antheridia and archegonia. The sperm will then fertilize the egg creating a zygote, and then the zygote will then turn into a sporophyte and a new plant will grow from it. After that, the cycle will then repeat. Another example of the earliest vascular plants is Aglaophyton major. As we can observe, it now has branching structures, has sporangia that produces spores at the tip, and has rhizoids that anchors it to the ground. In the picture, it also shows a fossilized stoma. So stoma are pore-like structures that can be found in plants that are responsible for gas exchange. We will now be proceeding to transport system and evolution of roots and leaves. The xylem transports water and minerals while the phloem distributes sugar throughout the plant. These tissues allow the plants to grow taller and stronger. In terms of evolutions, plants have also evolved and developed roots for anchorage and absorption of nutrients, and the leaves for photosynthesis. Leaves evolved in two types. Microphyls are small and simple leaves. It has a single unbranched vascular strand, while megaphyls have larger and more complex leaves and have branched vascular systems. It is said that megaphyls are more efficient in photosynthesis due to their larger surface area. We will now be talking about the reproduction of seedless vascular plants. Seedless vascular plants reproduce using spores. Seedless vascular plants reproduce using spores. Specialized leaves called sporophylls bear sporangia, and there are two types of spore production. Homosporous plants produces one type of spores, while heterosporous plants produces two types, which are the megaspores and the microspores. Megaspores develops into female gametophytes produced in megasporangia on megaspores develops into a female gametophyte produced in megasporangia on the megasporophyllis, while the microspores develops into a male gametophyte produced in the microsporangia on the microsporophyllis. All seed plants and some seedless vascular plants are heterosporous. So now we'll be talking about the classification of seedless vascular plants. Seedless vascular plants are divided into two main groups, the lycophytes and the monilophytes. The lycophytes are one of the oldest groups of vascular plants, and they include club mosses, spike mosses, and quillworts. They have small leaves called microphylls and reproduce through spores. Some produce only one type of spore, while others produces two. Their gametophytes can grow above or below ground and sometimes form a symbiotic relationship with fungi. The monilophytes, on the other hand, includes ferns and horsetails. Ferns have larger leaves called fronds, and young fronds are coiled structures known as fiddleheads. They produce spores in clusters called sori and usually found in the underside of leaves. Ferns has both sporophyte and gametophyte stages, with the sporophyte being dominant. Major groups of seedless vascular plants. As discussed earlier, lycophytes are among the oldest lineage of vascular plants, and they include club mosses, spike mosses, and quillworts. The example of these are shown in the images, and lycophytes grow in tropical forests, often as epiphytes, which are plants that grow on other plants but are not parasitic. They have small leaves, and their reproductive structures are called sporophyll, which are often grouped into clone-like clusters known as trobili. Next is the monilophytes, which includes ferns, horsetails, and whisk ferns and relatives. Ferns has large leaves called megaphylls, commonly known as fronds. Horsetails belongs to the genus Equisetum, and whisk ferns and relatives represent simple vascular plants with reduced structures. Lastly, significance of seedless vascular plants. Seedless vascular plants played a major role in Earth's history. During the Carboniferous period, they formed vast swamp forests dominated by lycophytes, ferns, and horsetails. We can see an example of this in the picture attached. Over time, dead plant materials accumulated and formed coal deposits. They also helped reduce carbon dioxide levels, contributing to climate changes as a global cooling. Today, seedless vascular plants continue to play an important ecological roles. They provide habitats, prevent soil erosion, and contribute to nutrient cycling. These plants represent a major evolutionary advancement, allowing plants to grow taller, spread widely, and shape Earth's ecosystem.
And with that being said, that concludes our reporting. Thank you so much for watching.